Welcome back to the Deep Dive, where we take complex scientific literature, peel back the layers, and hand you the synthesized knowledge you need to be quickly and thoroughly informed.
Today, we are looking at a really revolutionary turning point in biomedical research. For decades, the industry has relied on two main tools: simple, flat, two-dimensional cell cultures and animal models.
And both of those, while they were foundational, have some, let's say, critical limitations that have really slowed down drug discovery.
Dramatically slowed it down.
Absolutely. And the future isn't just about doing research faster. It's about doing human-relevant research. And the tech driving this shift is, well, it's miniature, it's physiological, and it's incredibly intelligent.
It is.
Our subject today is the revolution being driven by mini organs, these incredible complex three-dimensional cellular clusters grown right in a lab dish. We're specifically zeroing in on human intestinal organoids, or HIOs.
And our sources for this, it's a fascinating stack of cutting-edge studies, market analyses, regulatory updates. They all detail how these models are being built, the technical hurdles researchers are jumping over, and, their explosive impact.
Right. So our mission here is to synthesize all this so you can get a deep, thorough handle on where things stand right now. And crucially, we're going to focus on how advanced tech like high-throughput imaging, organ-on-a-chip, and AI is making them go from the academic bench to the commercial pipeline.
The stakes here are they're profound for global health, for the drug industry. IOs are moving from this interesting academic toy to an essential clinical platform.
A direct line to patient-specific drug testing.
Exactly. It's poised to accelerate therapies and just fundamentally change how we look at a drug's safety and efficacy.
Okay, let's unpack this. The rise of the mini-gut and why it's poised to revolutionize personalized medicine.
Yeah.
So let's start at the very beginning. When you hear organoid, it sounds like a tiny cloned organ. But what is it really on a biological level?
Okay, so an organoid is technically an in vitro three-dimensional cellular cluster. They come from either adult stem cells from a tissue biopsy or from pluripotent stem cells.
In that second category, pluripotent stem cells, that includes iPSCs, right? Induced pluripotent stem cells.
It does, and that's critically important because you can generate those from an individual patient's own cells, like a skin cell, and then reprogram it back into a stem cell state.
And the magic, the real innovation here, isn't just that they grow in 3D. It's that they have this built-in ability for self-renewal, and I find this amazing, self-organization. Does that mean they just know how to build themselves?
That is precisely what it means. You give them the right environmental cues, the right scaffold to grow on, and these stem cells will differentiate and organize themselves.
On their own.
Completely on their own. They faithfully mimic the native intestinal tissue, complete with all the folds and cavities. They're just following their own genetic programming to rebuild that classic cryptovillus structure.
So when we talk about them being superior, we have to look at what they're replacing. For a long time, as you said, there were two main models, both with major flaws. Let's talk about the animal models first.
Right. Animal models, especially genetically engineered mice, they were the gold standard for testing things in vivo. But they have fundamental problems. First, just species differences.
A mouse isn't a tiny human.
Not at all. A mouse's inflammatory response is just inherently different from a human's. And that's why drug predictions, especially for really complex human diseases like inflammatory bowel disease, they so often fail to match up with what happens in a human clinical trial.
So you've got high costs, major ethical questions, and at the end of the day, pretty poor predictive power.
Yeah.
What about the other workhorse? The 2D cell lines, like CACO2.
CACO2. It's an intestinal cancer cell line, and we've used it for decades because it's cheap, it's easy, it's fast, but it is fundamentally flawed for trying to model real physiology. Well, imagine trying to model a vibrant 3D city using just a flat 2D map. You miss all the vertical complexity, the layers, the connections.
And what are these 2D monolayers specifically missing that the organoids are able to capture?
They're missing the physical environment. They don't have those complex cell-to-cell interactions. They don't develop true polarity, the top and bottom of the cell, which you need for transport. And they often fail to form functional tight junctions or a realistic mucus barrier.
And without those things, polarity, tight junctions, mucus, you can't really model absorption or infection properly.
You can't. You're missing the key architecture.
So the organoid solves this by being a detailed 3D replica Just how detailed is the cellular complexity inside these mini-guts?
The complexity is, it's remarkable. Intestinal organoids contain all the cell types that come from the LGR5 plus stem cells, which are the engine of renewal in the gut.
So those are the master cells.
They are. And
Yeah.
when they differentiate, you get a full team of specialized cells doing their jobs inside the organoid.
What kinds of jobs are we talking about?
Well, first you have the transit amplifying cells, the rapidly dividing ones. Then you get the fully differentiated cells. You have enterocytes, which are your main absorptive cells, critical for nutrient and drug uptake.
Okay.
Then you have goblet cells, which secrete mucus. That mucus barrier is your first line of defense, so you absolutely need them for infection models.
Right.
Then there are Paneth cells. These are vital. They sit at the base of the crypts and secrete all the niche factors, like Wnt3a and EGF that keep the stem cells happy and control local microbes. And finally, you have the enteroendocrine cells, which make hormones and connect the gut to the rest of the body.
It really does sound less like a collection of cells and more like a functional ecosystem.
That's a perfect way to describe it.
And this leads to something I find fascinating, this idea of regional fidelity. Does an organoid that you grow from the small intestine actually know it's from the small intestine and act like it?
This is truly one of the greatest aha moments in the field. They absolutely retain regional specificity. If you take tissue from the duodenum, the jejunum, or the colon, the organoids you grow will maintain unique, functional, and even transcriptomic signatures of where they came from.
So why should our listener, who's maybe focused on drug discovery, care so much about this spatial fidelity?
Because the gut isn't one uniform tube. Disease happens in specific places, and drugs get absorbed in specific places. The duodenum, for instance, is where a lot of first-pass metabolism happens. The ileum is for bile acids. The colon is a totally different world with its microbiome.
So if you're making a drug for Crohn's disease, which often hits the ileum, you need a model of the ileum, not just a generic gut model.
Precisely. If you're testing an oral drug, you have to know if it will be absorbed in the small intestine, or it needs to survive all the way to the colon to be activated by bacteria. This segment specificity dramatically increases the chances your preclinical data will actually mean something in a human. Without it, you're just guessing.
Growing these complex structures obviously requires a very controlled synthetic environment. Let's talk about the engineering behind that, starting with the physical foundation. The scaffold.
Right, the essential scaffold that's been used since the beginning is matrigel. It's a gelatinous protein mix that gives the cells the structural support they need to grow in 3D and self-assemble. It's like a temporary extracellular matrix.
But the sources we looked at highlight some pretty big drawbacks with matril, especially for scaling up. What are the issues?
The problem is really twofold. First, it's animal-derived. It's harvested from mouse tumors, which brings in contaminants and regulatory issues. But maybe more importantly for research, it has severe batch-to-batch variability.
So the composition is never exactly the same.
Never. The stiffness, the growth factors, it's always a little different. And if your scaffolding changes every time you run an experiment, well, your reproducibility just goes right out the window.
So the future has to be moving away from that, towards something defined.
And that's the hydrogel revolution. Researchers are really pushing into both natural and synthetic hydrogels as alternatives. The benefit is total control. These have well-defined chemical compositions, so you get zero batch-to-batch variability.
And it sounds like it's not just about structure. You can actually tune the environment.
Exactly. With hydrogels, you can flexibly modulate the biophysical signals, things like stiffness, porosity, that directly tell the stem cells what to do. You can engineer the stiffness to mimic the small intestine or the colon, creating a much more accurate and controlled niche.
Okay, now let's talk about the liquid these tiny organs are bathed in the culture media. You mentioned it's complex. It has to tell the cells to stay young, but also to specialize.
It is incredibly complex. The standard is something called LWRN conditioned medium, which simulates the natural stem cell niche. And it's not just nutrient soup. It's a cocktail of signaling molecules.
What are the key ingredients in that cocktail?
The absolute vital ingredient is WNT3A. WNT signaling is the primary fuel for intestinal stem cells. Without a high level of WNT, the stem cells just differentiate and the organoid dies off.
Okay, so that's the stay young and divide signal.
Right. Then you have EGF, or epidermal growth factor, which pushes for general proliferation and expansion, and things like gastronide for overall epithelial health.
But it's a balance, right? You need renewal, but you also need them to differentiate into all those cell types we talked about.
Precisely. So to control that balance, the media also contains specific inhibitors, things like A83SU1 and SP20-SERL2190, which modulate other signaling pathways. These are crucial for maintaining that delicate balance between proliferation and differentiation. You're basically tricking the stem cells into thinking they're still safe in their little crypt.
This complexity is also a challenge. I mean, the traditional 3D organoid has an accessibility problem, right? The important surface, the apical side that faces food and microbes, is turned inward.
It's a major engineering hurdle. The lumen is enclosed, so it's extremely hard to apply drugs or introduce bacteria. If you can't reach the target surface, you can't model absorption or infection reliably.
So how have scientists tackled that?
There are three main strategies. The first and most common for high-throughput testing is to basically sacrifice the 3D geometry for a 2D monolayer format.
So you flatten them out.
You do. For large-scale screening, you dissociate the 3D organoids into single cells using enzymes and then plate them onto a coated surface, like a 96-well plate.
But if you flatten them, how are they any better than the old CACO2 models?
Ah, that's the key. These monolayers, when they differentiate, are confluent, and crucially, they are polarized. They still have functional tight junctions and all the diverse cell types. You see stem cells clustering in one area, differentiated cells in another. The genetics still drive them to maintain that critical architecture, even on a flat plane.
I see. So you get the cellular complexity of an organoid with the accessibility of a monolayer.
Exactly. It allows for highly relevant transport and permeability studies that CACO2 just can't touch.
What's the second strategy for the accessibility problem?
The second is the reverse polarity model. If the enclosed lumen is the problem, you flip the organoid inside out.
So the apical surface faces the outside.
Right. It creates an apical outward orientation, which lets you directly apply drugs or microbes to the surface you care about. It's incredibly elegant for studying things like bacterial interactions.
That sounds like a great fix, but I imagine it's challenging.
It is. Maintaining stability and getting high proliferation rates is tougher, but it gives you the most biologically relevant interface for those interaction studies.
Okay, that brings us to the biggest missing pieces. What isn't naturally generated in the dish with a standard organoid?
Standard organoids are really just the epithelium. They completely lack a functional mesenchymal structure, they don't have vasculature blood vessels, and they have no immune cells.
And for modeling complex diseases, that's a huge problem.
It's a deal breaker for things like inflammation, fibrosis, or metastasis. You need blood flow to deliver drugs. You need immune cells to fight infections or drive inflammation.
So researchers have to engineer these missing parts into the system. Let's start with vascularization. Why are blood vessels so crucial, especially for cancer?
In cancer, the vasculature is the tumor's lifeline. It promotes metastasis, it drives drug resistance. So to model this, researchers use co-culturing. They'll add endothelial cells, or ECs, to the organoid culture.
And they just form vessels on their own.
If the conditions are right, yes. They spontaneously organize into functional, perfusable microvessels that wrap around the organoid, giving it its own tiny blood supply network.
Incredible. And what about the immune system? That's obviously critical for everything from IBD to immuno-oncology.
The immune compartment macrophages, T cells, dendritic cells, all has to be engineered in. For immuno-oncology, you need T cells to see if they'll attack the tumor. For autoimmune disease, you need macrophages. The best technique is co-culture, ideally using autologous immune cells.
Autologous, meaning from the patient themselves.
Exactly. If you're testing an immunotherapy for a specific patient, you need that patient's actual immune cells interacting with their own tumor organoid. It allows for incredibly precise screening, something animal models are just terrible at.
This blending of multiple engineered tissues, it sounds like we're moving towards something even more complex than a mini organ.
If we connect this to the bigger picture,
Yes.
we're moving beyond single organoids. The ultimate goal is creating assembloids.
Assemblages.
It's the fusion of different tissue components, epithelial, vascular, stromal, immune into one functional unit. Disease is a multi-tissue problem, so you need a multi-tissue model. An assembloid can mimic the complex tumor microenvironment, or TME, and that makes it a much, much better predictive platform.
Okay, let's pivot from the engineering to the actual utility. What are these mini organs letting researchers and clinicians do right now? Cancer seems to be leading the charge.
Absolutely. The main application is through patient-derived organoids, or PDOs. These are grown directly from a patient's tumor cells, often from hard-to-treat metastatic cancers. And their value is that they are a direct replica. They have the same genetics, the same structure, the same functional quirks as the original tumor.
The big question for a clinician has to be predictive power. If a drug works on the PDO in a dish, How often does that actually hold up in the patient?
The data is it's overwhelmingly positive. Studies on metastatic colorectal cancer, for example, tested standard chemotherapies on PDOs. They found a statistically significant correlation between how the organoid responded and how the actual patient responded.
And this isn't just a coincidence, it's tied to real outcomes.
It is. It's demonstrably associated with critical outcomes like progression-free survival and overall survival.
The sources mentioned some specific numbers on accuracy. What kind of rates are we talking about?
We've seen really impressive rates, especially for tough cancers like metastatic, gastric, and colorectal. The predictive accuracy has been reported as high as 79% to 88%.
Wow, 88%.
To put that in perspective, if a PDO test says a drug is going to fail, there's an 88% chance it will fail in that patient. That is a massive improvement over older models. And genetically, they've found that over 90% of the mutations from the patient's tumor are faithfully kept in the organoid.
So beyond just predicting if a drug will work, do they help us understand how the disease works? The mechanism?
Yes, the mechanistic insight is invaluable. They're perfect for studying signaling pathways. Take notch signaling, for example, which is a major player in the gut. Researchers can use organoids to turn that pathway on and off and just watch what happens.
And what do they learn from doing that?
Well, they've shown that just activating notch isn't enough to cause cancer, but if you combine it with shutting down P53, a really common mutation, it kicks off the epithelial mesenchymal transition, or EMT.
Which is a key step in metastasis.
It's when stationary cells become migratory. The organoid lets you dissect that exact molecular partnership that lets the cancer take hold and spread.
You could never get that level of detail in a living patient. Let's shift from cancer to gastrointestinal health in general, like inflammatory and infectious diseases.
Organoids have become a go-to model for IBD or inflammatory bowel disease. When you grow organoids from IBD patients, they actually retain the disease phenotype. They're smaller, their polarity is abnormal, and critically, they have fewer of the proteins that form tight junctions.
Wait, so is that defect in the epithelium just a result of the inflammation, or is it an intrinsic part of that person's tissue?
That's the million-dollar question, and the organoid answers it. Researchers found that these defects in tight junction proteins persisted even after they took away the inflammatory signals and grew the organoids in a normal, healthy medium.
So it's baked into the cells themselves.
It suggests the organoid is capturing a fundamental intrinsic epithelial problem, not just a reaction, and that completely changes how you'd approach designing a drug for IBD. You can target the root defect, not just the inflammation.
And for infectious diseases.
They're fantastic. They've been proven for pathogens that hit the gut lining, like rotaviruses, norovirus, and bacteria like H. pylori and E. coli. The 3D structure is perfect for watching that initial infection process unfold.
I remember one of the sources had a really compelling example about visualizing H. pylori. Could you walk us through that?
It's a great example. H. pylori is the bacterium behind most stomach ulcers. Using gastric organoids, researchers could watch how the bacterium attaches with incredible detail.
And what did they see with the live cell imaging?
They saw the bacteria didn't just attach randomly. Using confocal microscopy, they showed that H. pylori actually prefers to stick to certain depressed cells within the organoid. And even more specifically, they showed the bacteria were actively swimming toward a chemical signal released by the host cells.
So the host cell is unknowingly putting out a come here signal that the bacteria follow to their target.
It's pure biological warfare, and you can watch it happen in the dish, which means researchers can now try to block that signaling pathway to stop the bacteria from ever reaching their target.
Let's pivot to pharmacology. We know that when you take a drug, your body has to absorb, distribute, metabolize, and excrete it ADME. How are organoids changing that field?
They are central to advancing ADME studies. The intestinal epithelium is that critical first checkpoint for any oral drug. And human intestinal organoids are proving to be massively superior to old models like CACO2 for studying this.
What was the big limitation with CACO2 for this kind of work?
CACO2 is a cancer line, and it just lacks the right concentration and function of key proteins. Compared to healthy human intestinal organoids, CACO2 shows much lower activity of crucial drug transporters.
And these transporters are like molecular bouncers at the cell door.
Exactly. Two critical ones are P-glycoprotein, or PGP, and DCRP. PGP is an afflux pumpet, actively pumps drugs out of the cell. If your model has low PGP activity, you'll way overestimate how much drug is absorbed, which can lead to clinical failures. Organoid monolayers have high human-relevant levels of these pumps.
What about metabolism?
Organoids also show significantly higher metabolic activity, especially from the CYP3A4 enzyme. That one enzyme metabolizes about half of all drugs on the market. Getting an accurate read on it in the intestine is essential for predicting the first pass effect.
So you can literally watch this transport happen in real time.
You can. It's really elegant. Certain drugs get absorbed along with protons, which makes the inside of the cell acidify for a moment. You can visualize this immediately with fluorescent probes.
So the cell's interior literally changes color when the drug goes in.
Precisely. They show that when you expose duodenal organoids to an antibiotic like cefadroxil, you see this immediate intracellular acidification. It's direct, dynamic, functional proof that the transporter is working.
And finally, we all know drug responses vary wildly between people, inter-individual variability. How do organoids help with that massive challenge?
This is really the pinnacle of personalized medicine on this platform. It's the power of iPSC-derived organoids. Since they come from a patient's own cells, the resulting organoids have that patient's specific genetic makeup, including any variations in their drug-metabolizing enzymes.
So you can create a mini-gut that metabolizes a drug exactly like its owner does.
Exactly. This lets you evaluate differences in CYP activity across different people due to genetics, sex, or age. By plugging that patient-specific data into pharmacokinetic models, you get crucial insights into why responses vary. You can start refining dosing for subpopulations before you even start a clinical trial.
So we've established the biological superiority, but to really change R&D, these things have to be scalable and fast. This brings us to the tech ecosystem automation and imaging.
Right. Organoids are more predictive, but they're also way more complex to handle than a flat cell line. To screen thousands of compounds, you need full high-throughput screening, or HTS, and that requires a lot of tech.
Walk us through what that automated pipeline looks like.
It starts with robotic sample processing, media changes, plating, all of it. But the real bottleneck is analysis. You can't just use a simple plate reader on a 3D structure.
This is where high-content imaging comes in.
Exactly. These are basically automated confocal microscopes built for speed. They can grab detailed images from hundreds of wells at once in a 96-well plate format. They can get all sorts of info at the same time: cell shape, fluorescence, how the cells are reacting.
And once you have all those images, you need the software to make sense of it.
Absolutely. The pipelines use powerful image analysis software to do things like separate the nucleus from the cytoplasm and quantify fluorescence. They use normalization techniques to get scalable, unbiased data, which is essential for quantifying complex changes quickly and accurately.
The next big leap in engineering is creating a dynamic environment, the organ-on-a-chip, moving the mini organ from a static dish to a living system.
OOC uses microfluidic channels on a polymer chip to create a microphysiological system. This lets you precisely control the environment in ways you just can't in a static plate media flow, nutrient gradients, and crucially, mechanical forces.
And the gut-on-a-chip is the flagship example. What can it do that a static culture can't?
It adds dynamic movement and a controlled atmosphere. It can sustain a complex human gut microbiome, which needs anaerobic conditions right next to the human cells. And the chips are designed to stretch and contract, simulating peristalsis.
Why is simulating peristalsis so important?
Because that stretching and fluid flow are active signals. They influence gene expression and tissue organization. Without them, you can't accurately model inflammatory conditions or host microbiome interactions. It's just not biologically relevant otherwise.
So if one organ on a chip is good, linking them together must be the ultimate goal.
And that gets us to the MOOC, or multi-organ-on-chip system. This is where you link chips representing different organs, intestine, liver, kidney, with circulating fluid that acts like synthetic blood.
So you can model systemic diseases.
Right, like cancer metastasis. That's a multi-organ process. A tumor cell leaves the colon, circulates, and then specifically colonizes the liver or lung. MOOC models can simulate that entire journey, and they even show organotropism.
What's organotropism?
It's the tendency of tumor cells to prefer certain secondary organs. Researchers have used link systems to show that tumor cells tend to stick to the lung chip because it expresses high levels of a specific chemical signal, a chemokine.
Like a beacon drawing them in.
It's exactly like a beacon. By linking the organs, you understand the systemic signaling that drives the whole disease.
And finally, we get to the thing that synthesizes all this data. Artificial intelligence.
AI is the bridge that turns all this raw biological data into predictive clinical insights. AI and machine learning are used everywhere from automating image analysis to rapidly analyzing complex readouts.
But the real power comes when you plug that high-quality organoid data into computational models, especially for pharmacokinetics, or PK.
What's fascinating here is how AI is revolutionizing PBPK models, physiologically based pharmacokinetic models. These are mathematical simulations that predict how a drug will move through the body.
And traditionally, they were based on uncertain animal data.
Exactly. But now you replace that uncertain animal data with highly accurate human organoid data. You feed the model real human intestinal permeability, real human liver metabolism data. This dramatically improves the prediction of what will happen in a human.
So the practical benefit is refining dosing strategies with much greater certainty.
It is. It minimizes the need for those expensive dose escalation studies in early trials, which saves a huge amount of time and money, and it flags potential risks way earlier in the process.
And we are seeing real-world successes from this organoid plus AI platform already.
A phenomenal example is the world's first drug candidate discovered and designed using this integrated platform. It's a drug for diffuse gastric cancer called SIG-Xdarin-94R. The platform wasn't just used to screen, it was used to design the molecule.
And the result?
It got FDA Fast Track designation and raced to Phase I clinical trials in less than 4 years. That kind of timeline is it's unheard of in traditional drug discovery. It perfectly shows the power of combining these technologies.
Okay, so the tech is there, but pharma is a slow-moving giant, heavily regulated. For organoids to really take over, they need regulatory buy-in. Is that shift actually happening?
It is happening, and it's happening decisively. The main catalyst was the FDA Modernization Act 2.0. This law explicitly promotes using what are called new approach methodologies, or EMS, which includes organoids, as valid alternatives to mandatory animal testing.
So that's a signal of intent. But what's the practical impact? Is the FDA actually starting to accept human-based data over animal data?
That is precisely the direction. It signals a phased reduction in animal efficacy testing. The NIH is actively encouraging the industry to adopt these human-relevant approaches because the goal isn't just safety anymore, it's human-relevant safety and efficacy.
That's a key phrase. But encouragement is one thing. Do we have a concrete precedent? Has the FDA actually approved a clinical trial where the proof of efficacy came only from these models, completely skipping the animal studies?
Yes. And this is probably the biggest regulatory headline in the whole field. There was a recent landmark achievement, the world's first FDA approval for an investigational new drug, where the efficacy data was generated solely from human vascularized organoid studies.
That is huge. What does that mean practically?
It means the data from the organoids was considered sufficient to provide the required proof of concept. It eliminated the need for traditional animal efficacy testing, which has been a bottleneck for decades. It's a milestone that shows regulators are accepting this human-relevant data as predictive enough to move straight to human trials.
So if the regulatory hurdles are coming down, the commercial money must be following.
The sheer scale of the investment confirms it. Just look at the infrastructure. The market for intestinal organoid culture medium alone, that complex cocktail we talked about, is projected to hit over $4.5 billion by 2030.
And that's just the media.
That's just the media. And that value is being driven almost entirely by the demands of pharma and biotech companies.
So who is stepping up to provide these services on an industrial scale?
The major contract research organizations, or CROs, are all launching dedicated high-throughput organoid services. Samsung Biologics, for instance, is focusing heavily on using patient-derived organoids for screening new anti-cancer drugs. They know their clients are going to demand this data.
And this is more than just setting up a few dishes. This is industrializing a biological process.
It involves massive investment in automated, scalable production. They're developing their own ECM substitutes to get away from Matrigol, designing custom culture containers, and building fully automated, closed-system facilities that can run hundreds of thousands of cultures at once.
So the industry view is clear.
It's crystal clear. As the FDA shifts away from unreliable animal testing, these human-relevant models are emerging as the only practical, realistic, and highly predictive alternative. It's not a niche technology anymore. It's becoming a foundational necessity.
We have covered a tremendous amount of ground today. We've traced the journey of the human intestinal organoid from a simple stem cell cluster into this sophisticated multi-component dynamic system, an assembloid, all powered by automation and AI.
And the core of this whole revolution is the ability to culture and analyze complex, patient-specific human tissue in vitro. It captures the unique traits of an individual's disease. And that has moved these tools from a fascinating academic curiosity to a clinical necessity.
The relevance for the drug pipeline is just impossible to overstate. We know about the 90% attrition rate in clinical development, mostly due to poor models. By giving us data that is orders of magnitude more predictive, HIOs offer the clearest path yet to finally chipping away at that massive failure rate.
Which, it raises an important question for the future. We've seen that integrating patient-specific organoid data into these computational models can already predict how individuals will respond to a drug.
Yeah.
As multi-organ-on-a-chip systems get even more complex, mimicking a virtual human, how will this technological capacity redefine the ethical and even the logistical necessity of traditional clinical trials in the next decade, especially with the FDA already shifting its stance?
A profound thought to mull over as the science fiction of building a human body in the lab becomes the standard medical practice of tomorrow. That was your deep dive into the intestinal organoid revolution. We'll see you next time.